our last talk of the seminar series, and thank you very much for everyone who's hung around, and thank you to Chris for being our last speaker. We have Chris Langsdorff from Thermo Fisher Scientific, and the talk title is Studying the Immune Response to Viral Infection with Flow Cytometry. Chris, you can go ahead and share your screen.
Thanks, Tina. Can you see that okay?
Yes, we can.
Great, and I guess that means you can hear me as well, so we're all set. All right, thank you everyone for sticking around for Friday afternoon. It looks like we have a good group on the line, more than 80 people, so appreciate everybody hanging out for Friday afternoon. As Tina said, my name is Chris Langsdorff. I'm a staff scientist in our research and development group in Oregon. So currently working from home, as most of us are. You may hear the kids come in here in a few minutes to get lunch, but we'll try to carry on as we do. Normally my research is focused on immuno-oncology and developing fluorescence-based methods to study the interaction of the immune system with cancer. But as many of us have, we've undergone a pivot the last several months to begin looking more closely at how the immune system interacts with invading viruses in our bodies. And so that's really the focus of what I'll talk about today, is different methods focusing on fluorescence-based single-cell analysis, where that's typically flow cytometry, but that can also mean quantitative imaging, as we see in some of these images here. And so, as we've heard in the previous presentations, and we've seen a major change in our lives the last few months, and this has led to some significant challenges in how we live and how we work. But this significant change is also a source of innovation. It really gives us a reason to start, very, very quickly looking at new techniques and new ideas. And I would say innovation was once explained to me as not inventing new things, but looking at connections between existing ideas and creating new ways of thinking of things. So really what I hope to do today is show you some examples of techniques that can be used to study viruses and their interaction with our mammalian cells to hopefully stimulate your thoughts as you go forward with your research to answer some of our current challenges. So for today, I've split it into three focus areas. First, looking at how viruses actually enter into our cells and how we can monitor that with fluorescence. Then in the case of RNA viruses in particular, looking at how we can monitor the synthesis and replication of the RNA and the entire virus. And then finally, how our immune system responds to and reacts to those invading viruses, and how we can monitor that. So first, when a microbe is being taken up into one of our cells, we have a good fluorescence-based way to study this. As an introduction to this, I can show you how we can label E. coli, so bacteria in this case, with a pH-sensitive dye called Frodo. Now, when we add these E. coli to macrophages, as we see here a field of macrophages, if we add E. coli labeled with Frodo green in this case, we don't see green signal until we observe these cells over time. And over the course of an hour, we see these macrophages eating the E. coli, and the individual E. coli cells become very brightly green fluorescent. That's in response to the acidic environment in those phagosomes. So we can see in a schematic here, as one of those bacteria transitions from the neutral pH environment outside the cell into the acidic environment of a phagosome, this pH-sensitive photodye will become significantly brighter. And so this is something we can measure with fluorescence microscopy or with flow cytometry. And so we can look at the, the results of the same experiment on a flow cytometer and see that we have a background level of signal in untreated cells. When we load our cells with this labeled E. coli at 37 degrees, we get a really bright signal. And an important control in this kind of experiment is just to do the entire experiment on ice, or at 4 degrees as shown here. And in that case, the E. coli is able to bind to our cells. It's just not able to be phagocytosed. That's a temperature-dependent phenomenon. So just incubating on ice will knock this back down to 0. And so both of these experiments are just in a macrophage cell line. We can make this a bit more realistic by doing the same experiment in whole blood. And so if we take those same, that same E. coli labeled with the pH-sensitive photo-green dye, and we now add those to human whole blood, so this is fresh heparinized human whole blood, we can analyze this whole blood by first ignoring our erythrocytes. So we've labeled this whole blood with a dye called dicyclyl violet, as seen in step 2 here. And this will identify only cells with a nucleus. And so thus we've gated out all of our erythrocytes. So we can further analyze our nucleated cells and ask what is the brightness of those cells in this photo-green channel. And thus we see some cells did not phagocytose the E. coli and some did. If we backgate this positive population, we can see, in fact, it is our neutrophils and our monocytes that carried out this phagocytosis. It was, as expected, our lymphocytes are not able to carry out this process. And now I'll call out that we analyzed this experiment on an attuned flow cytometer. This is an acoustic focusing instrument that lets us analyze this whole blood at a really fast rate so that we can dilute the whole blood and then acquire our cells at a really fast rate. And so 1,000 microliters per minute in this example. So we can still get a significant number of cells to analyze, but in a whole blood environment. We're still talking about bacteria, but I had mentioned we should be looking at viral entry. So we can use the same class of pH-sensitive dyes. Here we'll use photo red to look at phagocytosis of viral particles. Here we're studying H1N1, and we've actually labeled the H1N1 virus with a dye called dye O. This is a lipophilic green dye, and so in the course of this experiment, our MDCK cells were treated with the indicated controls for 1 hour. Meanwhile, the H1N1 virus was labeled with a tracking dye, and then at the end of this hour, both the labeled virus and the photo red on a 10,000 molecular weight dextran were added to these cells and allowed to endocytose for 10 minutes. Now we can see in our untreated controls or vehicle controls that the spots, the punctate spots that show us where this virus has been endocytosed are also acidic, as indicated by the photo dye. If we use various neutralizing or alkalinizing treatments like ammonium chloride or baphylamycin, this pH dependence is, low pH environment is neutralized. And so we can, in fact, report that this H1N1 virus is internalized into acidic organelles. We can do this with flow cytometry also. And here, instead of labeling the virus with a tracking dye, we're actually directly labeling it with an amine reactive version of this photo red ester. Here, the IFL is really for improved fluorescent labeling. So if we're labeling larger particles like viruses or antibodies or bacteria, this new IFL version of Frodo is more soluble, and I'd highly recommend it for this type of experiment. So in this case, reovirus was labeled with our amine reactive version of the pH-sensitive Frodo, and then we add that dye to our HeLa cells for 1 hour on ice. This just lets the virus bind to its specific receptor on the cell surface. After that, we'll wash unbound virus away, and then incubate at 37 degrees for the indicated times. And at the end of each time point, we'll analyze those samples with flow cytometry. And so we can see a couple of things. First, over time, the medium fluorescence intensity of our individual cells in this Frodo channel, which in this case, this we read in a channel like our RPE channel, we can see that signal is significantly increased. If we again treat with an alkalinizing treatment, ammonium chloride, a fairly high dose in this case, we knock that signal back down to baseline. And so we're actually directly reporting the uptake of these viral particles into single cells based on their acidic environment in those endosomes. Now, we can move on now and look beyond viral entry to what happens once a virus is inside a host cell. We've seen a lot of reports in the news and in the literature of different drugs aiming to be used for our current COVID-19 and SARS-CoV-2 virus. And so imagine we've all seen remdesivir described. This is Gilead's drug. It's actually a prodrug, which allows it to enter into our cells and then be metabolized. And it's actually this active metabolite that carries out the antiviral function. Now, this is a nucleoside analog, actually an adenosine analog, which once the now I suppose when RNA polymerase is carried out, or RNA synthesis is carried out by an RNA polymerase, that polymerase will incorporate whatever nucleoside bases are in its environment. If our cells or our patients are treated with remdesivir, that RNA-dependent RNA polymerase that the virus relies on will incorporate this compound. Once 3 of those are incorporated, that replication will terminate, and we have the expected prevention of replication of our virus. Now we can use a similar approach, that is an artificial nucleoside, to actually measure the replication of our viruses. So instead of adenosine, let's switch to uridine, which will be incorporated, this ribose group will be incorporated into an extending RNA strand, in the case of the virus by the RNA-dependent RNA polymerase. Now we can add an artificial tag to this uridine, that is a carbon-carbon triple bond, that is an ethynyl group to this 5 position, and we can feed this compound either to our cells or to whole animals that we're interested in studying viral replication in. Now, if we feed this compound to our cells, this will now be incorporated into every nascent RNA strand, meaning that we can then interrogate our cells for those that now contain this tag. And to do that, we use click chemistry, specifically using this Invitrogen Clickit Alexa Fluor RNA assay, which provides all of the reagents required, both the EU, as we call this, the components required for the click reaction, and an Alexa Fluor dye azide. Now, this azide is another very small tag, And both this and the ethynyl group are bioorthogonal. That means they shouldn't exist anywhere else within our cells. So this is a very specific reaction. And so we'll get a very sensitive and specific indication of where RNA is being synthesized in our cells. Now, on the right, we see an example of how this works in a mammalian cell line in just 3T3 cells. So here, the EU was added for an hour at 1 millimolar, followed by a fix and perm step, and then this click reaction. We can see our green is our tubulin counterstain. In blue, we see the nucleus. And then the magenta shows us the location of RNA synthesis in these cells. Now, of course, we'd expect a very substantial amount of RNA synthesis in these actively proliferating cells. That might actually be more signal than we would see than if these cells were also infected with virus. And so we have controls we can use for that. One is to treat our cells with actinomycin D, which essentially blocks genomic RNA synthesis. So in this case, we switch models and look at synthesis of coronavirus RNA in mammalian cells. So the top left panel, we see what RNA synthesis from the host genome looks like. So we have a very large amount of RNA synthesis. If we treat with actinomycin D, that completely goes away. Now, if we include the viral infection in these cells and actinomycin D, we can see that nuclear staining is gone, and now we're only seeing the alexaflor 594 signal in the cells where the viral RNA is being synthesized. We can look at this over time as well, and we see 15 or 30 minutes post-infection, there's really no appreciable RNA synthesis. But then at 45 and 60 minutes post-infection, we really see substantial viral RNA synthesis. And we can say this is specifically due to the RNA-dependent RNA polymerase that's not affected by this actinomycin D. And so we can multiplex this assay, of course, here with a couple of antibodies, again looking at the RNA synthesis carried out by replicating a coronavirus in mammalian cells. And here we'll counterstain, first we'll feed the EU probe and then fix and perm and click on our alexaflor 488, and we can see the distribution of RNA synthesis due to viral replication in these cells. We can then use antibodies, that is fluorescently tagged antibodies, to label this non-structural protein 12, and then another antibody against double-stranded RNA. And again, we'll expect this double-stranded RNA to also show us the location of our total replicating virus, while in green we can see nascent RNA, and we can begin to look at the colocalization of these different reporters of coronavirus replication in host cells. And so this is valuable because we can multiplex, and it's a very specific and sensitive measurement of RNA synthesis. But one consideration here is that the coronavirus we're studying here doesn't just have one gene, it has multiple genes. And so what if we want to study the transcription of an individual gene? So to do that, we'll switch to a different technology. This is a hybridization amplification-based technology, where for imaging, we call this View RNA Cell Plus. And using this, we can actually detect the RNA of an RNA virus. In this example, that virus is Zika. And with no virus, we see minimal background. With infected cells, we can see these bright red spots indicating the location of single virions in these host cells. And this labeling is dependent on a single transcript, that is the VF1 19981 here, a single gene being expressed by that virus. And so for flow cytometry, this assay is called the prime flow RNA assay. And essentially what it means is we can do single cell quantitative analysis of a single messenger RNA or a microRNA in single cells. So you choose what RNA target you would like to study, and we'll provide you with two target probes that will hybridize specifically to this. And then through a sequential hybridization process, ultimately attaching fluorophores to this amplification tree, we are able to attach a very large amount of fluorescent signal to every transcript in our cells. And we can also multiplex this and look at multiple different transcripts in single cells, up to 4 different ones per cell, each with a different color of fluorophore and a different flavor of oligos that we're hybridizing on. And so how does this look in flow cytometry? As an example, we can look at the expression of, the transcription of HIV RNA in helper T cells. So through a standard gating strategy to give us live T cells and singlets, we can dive further and look at our CD4 positive helper T cells. And then using prime flow to target HIV-1 RNA, we can look at very rare reservoir cells that are actually expressing this HIV-1 RNA. So an uninfected individual, we'd expect a minimal background here. In a patient treated with antiretroviral therapy, again, we see minimal background. In untreated patients, we can see a percentage of helper T cells containing this HIV RNA that actually increases in relation to the copy number of viruses measured in those the blood of those patients. So we can actually pick up a very rare event using this technology. We can also multiplex this with antibody-based labeling. So we can switch and look at the another messenger RNA expressed by HIV, and also look at the protein produced by this messenger RNA. And we see a variety of different groups as a result of this dual parameter plot. Some cells that have RNA but no protein yet, some that have begun that translation into protein, and some that have a very significant amount of both the RNA and the protein. It's possible to sort all 4 of these cell types and actually image them using confocal microscopy and look at the localization of these fluorophores, showing us in red the protein and in green this HIV RNA within our helper T cells. And of course, our T cells are largely a nucleus and not much else. So we're confined to the periphery of these cells by imaging. So an interesting example of how to multiplex these is shown in the ability to create a different probe set against the positive strand of RNA and the negative strand of RNA. In this example, using yellow fever virus, we created a specific probe against the positive strand, which is present immediately after infection, and another separate probe against the negative strand, which only appears after the RNA has replicated in the host cells. These will each be tagged ultimately with a different fluorophore, and so by flow, we can visualize on the x-axis, or sorry, on the y-axis, the presence of positive strand RNA, which gives us a measure of infection. So we see after 2 days, we get a very significant amount of infection. But on the y-axis, we don't see that negative strand RNA appear really until day 3. And that gives us an indication of replication of the virus. So using one technology with 2 different probes, we can do a kinetic measurement of both infection and replication of this virus in our host cells. So finally, wanted to talk briefly about the effect on our host cells and the effect on our immune system. Now, this has been widely studied, especially in the last 3 months or so. The amount and the rate of publication is really impressive, with groups working really rapidly to understand better understand and treat our current COVID-19 situation. A, a, a recent paper from the group of Andrea Kosarica in Modena looked at the production of pro-inflammatory cytokines and compared both control healthy human donors to the blood of COVID-19 patients. And they found quite a significant increase in inflammatory cytokines, IL-2, interferon gamma, IL-17 in COVID patients as compared to controlled donors. If we quantify this data and assign significance to it, we can see, in fact, a very substantial increase, and in some cases a significant increase in these cytokine production on a single cell level. So here in cytotoxic T cells, we see a substantial increase in IL-17A and also in IL-2, ultimately recruiting cells like macrophages and neutrophils that are both responsible for the resolution of this viral infection, but also responsible for a lot of the cytokine storm that those patients experience. And in fact, the percent of cells expressing both IL-2 and IL-17, as we could call these polyfunctional helper T cells, is also very substantially increased. So this IL-2 and IL-17 co-producing cell is quite significantly increased. And it's not just the percent of cells producing cytokines that has changed. It's actually the number of different immune cell subsets in circulation that has changed in COVID-19 patients. And this group of Andrea Kosarica explained this very clearly. But we've seen other data. In fact, a very early publication in 2020, comparing the normal range of immune cell subsets in blood to those seen in moderate or severe COVID-19 patients. Here we're actually showing the cells per microliter. We can see in COVID-19 patients, both subsets, helper and cytotoxic T cells, the absolute count is quite significantly decreased, as are natural killer cells, while in fact the number of B cells in circulation is not substantially changed. A couple explanations for this were given. One is that these T cells are just undergone activation to cell death and are gone that way, but also we see them substantially leaving circulation and entering the infected tissues. Now, this measurement of cells per microliter can be made in a few different ways. We can use counting beads added to our sample to allow us to calculate the cells per microliter, or we can use a flow cytometer that directly provides us that measurement. And so in this case, we use the Attune-NXT flow cytometer. It's a syringe-driven system that gives us a direct volumetric measurement of cells per microliter. So no counting beads required. Just in our statistics window, we can actually indicate the concentration of individual cell types. So this is compatible with high throughput capabilities, 96 or 384 well plate loading, and then a full suite of automation. So whether you're screening for antibody binding or viral entry, we're able to meet your needs, whatever the scale is with this system. And a recent introduction, just this Q1 of 2020, is our Cytkick Max auto sampler that cuts the time for a 96 well plate down to 22 minutes. And so really substantially increasing the throughput by about threefold for these screening experiments. So
Just
it's,
let you know, oh, sorry, just want to let you know we just have just a couple of minutes left.
oh, thanks, Dina. Yeah, we're right on time. I want to make sure nobody misses. It's 3 o'clock on Friday in New York. I don't want to keep you from happy hour, but thank you everyone for joining us, and I really appreciate the invitation to present today, and thank you to all the organizers for including us in this seminar.
Okay, great. Sorry, I cut right through your last slide like that. I did not realize.
Nope.
Thank you so much, Chris, for such a wonderful talk. I think I'll ask just one question. We're you know, right at that 3 o'clock mark, but I think we have time for one question. So I was curious with the prime flow treatment, and we're looking at RNA by flow cytometry. I have some experience with, you know, looking at cellular RNA from the eukaryotic cells that we're looking at. But when we're talking about viral loads and viral RNA, has there been any testing to see if the fixation and permeabilization of the cells has any impact on the viral load? You know, are they sensitive? Is the viral RNA sensitive to the treatment that prime flow requires, and would have any impact on the levels there?
Yeah, you know, that's a great question. I think for that specifically, it's an important question, and I'll have one of our technical specialists follow up on it. But in general, it's the same fix and perm procedure we would use for intracellular antibody labeling, and so it's nothing harsh or unusual. So I think as long as things are fixed in place, I wouldn't expect the viral RNA to, you know, be washed out or be lost. Especially considering the viral RNA is, you know, these are large transcripts. It's not, if we're able to preserve microRNA in cells without it being washed out, I think it's not going to be an issue for viral RNA.
Okay, great. Thank you so much, Chris. That's all the time we have for questions today, and we really appreciate you joining us with such a wonderful talk. So thank you.
Great, thank you very much.